All right, y'all, let's work through exam number 1 together. All right. So let's work through exam 1. Okay, so this first one, I had these rock bunnies, and you can imagine that I could make the following observations, or I did make the following observations. When the bunnies are in the sun, they warm up. They seem to absorb energy from the sun, and I can feel them giving off energy as well. Okay, so this somehow seems like the E of decree, right? So I'm going to be thinking about decree for this question, right? So DNA, energy, cells, reproduction, evolution, and response to the environment. The other thing is that there seems to be a mama bunny and a baby bunny, so it seems like they've reproduced. After taking a sample from their backs, I looked for cells under a microscope and found cells of many shapes and sizes. So there's cells, but they're different, right? Lots of different ones. And the sample from their backs stained with a dye only sticks to DNA. That only sticks to DNA shows that there is DNA from the cells in the sample. So that also is the D, okay? So when I think about decree, right, we have some DNA, but I want to know, is it bunny DNA? Or from other organisms? Right? For the E, the energy, it seems like they're taking it in and giving it off, but are they actually using the energy? Right? Using the energy, or are they just holding it for a little while and then letting it go again? For cells, it says that there's many shapes and sizes, and I want to know if any of those cells are bunny. Are they bunny? Or maybe they could be microorganisms, right? So maybe they're prokaryotes or other microbes. Maybe some of my skin cells have come off on them. Who knows, right? Reproduction, it seems like they've reproduced with there being a mommy and a bunny, but I want to watch to see if they reproduce again. And then for evolution, no evidence of evolution. Okay, and then for responses to the environment, no evidence. Evidence that they're responding to their environment anyway. So here are some things that I want to know about. I would say the bunnies are probably not. And part of that reasoning is because it doesn't seem like they fill all of decree, right? So for life on Earth, all 6 of these are required for me to decide that something is living. And then also some of these things might be a little bit misleading with like things like the energy. Are they actually using the energy or just holding it and then releasing the same amount? Okay. So for the next one, we have labeling the DNA molecule below. So I have a deoxyribose sugar. Maybe I'll switch back to my other pen. So I have a deoxyribose sugar. So I'm going to put this right here is my sugar, right? A phosphate group, maybe I'll do a different one just to make it easy. Phosphate group here, right? A nitrogenous base, so that's like right here. A phosphodiester bond. So remember what a nucleotide is. So maybe it might be easier even if you had done a nucleotide first. So right here is a nucleotide, right? It includes a phosphate group, the sugar, and the base, right? All 3 parts. So that means that this line right here is the one that is the phosphodiester bond. Okay? That connects 2 nucleotides together. Okay? So phosphodiester bond. This right here is my nucleotide. Circled. One DNA strand is this whole thing here, right? And so you could have done this in a few different ways where you might have highlighted a whole strand, right? And so that this is a strand versus the entire molecule is this whole thing, right? 5' and 3' ends. So here's that OH group, right? So there's my 3' end versus over here. I'm gonna actually switch colors because I feel like I'm getting too much for one color. Okay, so I've got my 3' end here with the OH and the 5, so think 5-phosphate and has that same sound with it. 5' end over here, 3' end down here. I can check that I have these two as being anti-parallel and that on the same strand I have both a 3' and a 5' end, okay? So 5' and 3' ends, and then fill in all the missing bases. So remember, T's always pair with A's, C's always pair with G's. Oops, let's see. And then add all the missing hydrogen bonds. So 2 for A's and T's, 3 for G's and C's. Okay. Next we have Meselson and Stahl. So this is semi-conservative replication. So I wanted you guys to start by splitting this single molecule apart into its 2 separate strands. And then after the first round of replication, they gave cells nucleotides labeled with blue dye. So here I have my new ones, right? And then these, remember, split again. So I've got my red ones, I've got my blue ones that are all being used as a template for the new DNA, which in this case is going to be my yellow. Okay? And so what percentage of DNA strands have yellow? So 50%, right? So 1, 2, 3, 4 out of 8, 50%. And then what percentage of molecules have yellow after the second round? And that's 100%, because this is 1 molecule, 2, 3, and 4. All of them have at least some yellow after the second round of replication. You guys did really well on this next one. So if the top strand is the DNA template, and the GAGA sequence is the primer for DNA replication, you had to fill in the rest. So here's my 5' end on my top strand. So I'm gonna just fill in the rest of my 5' and 3' ends. You didn't have to do that. It just helped you to say like, oh, I'm gonna add to the 3' end. That's over here. So T, C, G, T, C, C, T, A. Okay? All right, so the next thing was to look at this cell on the right and see if it was in G1, S, G2, or mitosis. So here you can see that I have unreplicated chromosomes. And when I have unreplicated chromosomes, that's going to tell me that I'm before S phase, okay? So I'm going to be in G1, right? Because I've got homologous chromosomes, but they're all in, oh, those aren't homologous. That one and that one are homologous. And they all are just a single DNA molecule, okay? Okay, so I'm going to choose G1 because chromosomes are not replicated, right? So it's before replication. Okay, so next I'm going to draw the same cell at metaphase, right? And remember, this was when we only knew about mitosis, right? So mitosis, and the cell at the right. Okay, so I want to have all of these replicated. And remember, in metaphase, I'm lining up on that metaphase plate, right? So I'm going to have my big C, my, oops, the D is below the centromere, I think, the big D, the little d, and the other big C. You could have put them in any order here as long as they were lined up in the middle, and they were going to be being divided by their sister chromatids, okay? So done with that. How many chromosomes are in the cell in metaphase? So even though they're replicated, there are only 4 chromosomes, okay? How many DNA molecules are in the cell at anaphase? There's going to be 8. Remember that we're splitting these apart and pulling them apart at anaphase, so they're going towards the opposite poles, but it's not complete yet, okay? We haven't split the cell into two, so all 8 of these molecules, right, 1, 2, 3, 4, 5, 6, 7, 8, are all going to be in that cell. All right, the cell below contains 2 homologous chromosomes, and I wanted you to label one set of sister chromatids and one set of non-sister chromatids. So remember that sister chromatids are going to be identical and be part of the same replicated chromosome. Sister chromatids, oops, what was that? Okay. So these are sister chromatids, and then non-sister chromatids are homologous chromosomes that are part of different, or DNA molecules that are part of different homologous chromosomes, okay? So they're going to have the same gene. So if I had used that C gene, right, they're going to have the same genes. They could have different alleles, right? So here we have non-sister chromatids, okay, are these 2. Okay, how many DNA strands are in each sister chromatid? So a sister chromatid is 1 molecule, so there's 2 strands, okay? Describe the distribution of new and old DNA in each sister chromatid. So in each one, there's 1 old and 1 new in each sister chromatid. How are the sister chromatids made? I accepted a lot of different answers for this, but ultimately, somewhere in your answer, you need to say something about semi-conservative replication. Okay, and are the sequences found in the sister chromatids identical? And I really want you to explain your answer here. So the original, the strands in the original molecule are separated, and each are used as template. Okay. All right. Next was to label the image below with what stage of the cell cycle and mitosis they're in. You guys did really well on this. So this is an anaphase because they're being pulled apart. So think of ana, apart. So there's anaphase. This next one is metaphase. So think meta, middle. The next one, it looks like it's telophase. Like you would have to use a telephone to call, if some DNA in here wanted to call the DNA over here, you'd have to use a telephone because they're so far apart. This next one over here looks like it's in prophase. I think I also accepted G2 maybe or something. I might have accepted another answer as well for that, which wasn't quite as good, but I was okay with it. And then this last one, you could have either done G1 or after cytokinesis. I think I also allowed cytokinesis here. Telophase is better actually for this one, I think, but I was okay with cytokinesis as well. So the order depended a little bit on what you called some of these things, but for the most part, I was expecting you guys, let's say if it's the after cytokinesis is the one I'm choosing for that, it'd be 1, 2, 3, 4, and 5, right? If you're starting with G1 instead over in this cell, then you might want to have this one as number 1, right? Then 2, 3, 4, and 5. And I accepted either of those, okay? So next you want to define what each set of arrows is pointing to. So these are sister chromatids, right? They're going to have identical sequences, unless there's any mutation, right? These are going to be homologous chromosomes. These are going to be non-homologous. Chromosomes. These are non-sister chromatid. Right? Okay, and that was it for that one. Okay, so next RNA polymerase is moving left to right. See this arrow left to right. Is the X at the 5' or 3' part of the DNA? So remember that RNA pol, oh wait, sorry, I skipped. Oops, I skipped that first question. Okay, is the top or bottom the template strand? So the top is the template strand. It's the one being read. Right? is X at the 5' or 3' part. So remember, if we're moving left to right and RNA polymerase is going to read 3' to 5' because it has to write 5' to 3', right? Then I know that I've got this to be the 3' end. Oops, 3' end over here. X is going to be 3'. And then write out the RNA sequence. So if we're moving left to right, then my sequence must be behind me. Okay, so I would have a 5' end and then G, A, C, U, G, C, C, U, A, G, U, C, G, G, C, G, U, U. And then I was okay if you guys filled in the next few, or if you just stopped here, I was good with either way, okay? So you should have 5' to 3' already written, done that. This was the 3' end. And then label your RNA sequence. So I did the 5' end. This will be the 3' end over on this end. Okay. Next short answer 10. Gene A is expressed in the stomach and the skin, so in 2 spots. Gene B is expressed only in the heart. Add switches for both genes to the figure below. Okay, so gene A, I want a switch for the stomach and a switch for the skin. And then gene B is only in the heart, so I only want one switch in front of it. Will the same switches be found in the DNA in the brain and the DNA in the mouth? And explain your answer in 1 sentence. Yes. Switches are part of the DNA, and DNA is identical in all cells, except gametes. Okay, all somatic cells. Let's do that. Okay, how are genes A and B expressed in different parts of the body? Explain your answer in 2 sentences or less, but be specific. So the main thing here is activators. An activator for the stomach switch is only in the stomach. And will turn on, this is like an EG, for example, expression of A in the stomach, right? You could have said lots of things here. This is one answer. I accepted other things as well. Okay, next, gene C is expressed in the brain and nerve cells. So it wants me to draw a gene and label the promoter, transcribed region, the +1 nucleotide, and any switches. So I always think it's easier to draw the switches to the left and have transcription as if it was going left to right. So here's my promoter. Here I've got my transcribed region. Remember that the +1 nucleotide is the first nucleotide that is transcribed. And then I have 2 spots where it's going to be expressed. So I want 2 switches. So brain switch and nerve switch. Okay, draw all 3 proteins that you would find on the DNA in a nerve cell. Okay, so I would find transcription factors. So general TFs. I would also be able to find an RNA polymerase, right? And then I would also be able to find a switch activator for nerve switch. And you need to be specific that it was for the nerve switch, okay? So here I have an activator that only is for the nerve, okay? Okay, can you find gene C in a skin cell? Explain your answer. Yes. The gene is part of the DNA. Right? And remember that DNA is identical in all somatic cells. Can you find the RNA transcript and protein made from gene C in a skin cell? No. There is no skin switch for this gene. Okay. Next, label the gene below and make sure you include everything on the full checklist. Okay, so it looks like my start codon is over here and my promoter is over here. So the direction of transcription is going from right to left. That's the first thing that I want to do. Okay, so direction of transcription is right to left. So I know that my transcribed region has to start here. Transcribed region. I don't even know if it wants this, but I think it helps set it up because that tells me my +1, which is the first one that I asked for. So +1 site. Then the direction of transcription I already did. Here's my stop codon over here. My template strand. Okay, so if my direction of transcription is right to left, and it looks like here I have my 5' end labeled, so I'm going to add my 3', 5', 3', right? And so if it's right to left, and I need to read, right, 3' to 5', right? And so my arrow here is right to left, just like my direction of transcription. Then here is my bottom strand. It's going to be my template. And my top strand is going to be my coding strand. Okay, so template strand, the transcribed region I already did. A switch is right here. The coding strand is the top strand. Fill in the rest of the 5' and 3' ends. I did that. And then my UTRs. So my UTRs are going to be like looking at the coding strand, okay, is going to be 5' UTR. And 3' UTR. So this is closest to the 5' end on the coding strand, and this one is closest to the 3' end on the coding strand. Okay, and remember those UTRs are untranslated regions, right? So they're transcribed but not translated. All right, next is to consider the gene and its regulatory regions below. So it looks like there's 2 switches, so I'm going to have these expressed in skin and intestines. Must be epithelial or something. Okay, and here I have a promoter and then my transcribed region, so I have regulation left to right, and then next I'm just going to see what I want to add in. Okay, so next it says add in all proteins involved in transcription that you would find in every cell. So I'm going to have those general transcription factors again, right? You would also find the RNA polymerase in every cell. And that's all that I would have in every cell of the body, right? It's just those 2, okay? All right, where could a mutation occur that changes the expression of this gene in both the skin and the intestines? So here, it would be in the promoter, right? Because that would make it so that those transcription factors couldn't bind. And if those transcription factors can't bind, then I don't have RNA polymerase coming over, and then I have no expression. Where could a mutation occur that changes the expression only in the skin cells? So in the skin switch. Most of you guys got that one. Right, only in the skin switch would it change expression only in the skin. All right, and that's it. So that was exam number 1. I hope this is helpful as you guys are reviewing things and looking over previous content.